The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 11th episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcast, The Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, The Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam Cotty, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian Weil, and I've been in the flow cytometry CRO space for over 12 years.
Additionally, we have two guests joining us, our CEO, John Bucksath. John, do you want to say hi?
Yes. Good morning, gentlemen. Thanks for having me on the podcast.
And Stefan Cross from our partner lab at Crux in Australia. Stefan, did you want to say hi?
Hi, guys. Good to be with you.
Excellent. Thanks, guys. Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas with additional locations in Europe and now a partner site in Australia, we're both here in Philadelphia. Our producer, Jeremy, is in Missouri with John, and we have Stefan in Australia, as mentioned. So we are truly all over the map. We're overjoyed to have you listening to our 11th podcast, and it will be available virtually everywhere. Wherever you choose to find and play your podcast, you will likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to this or any of our other podcasts. Today's episode will be a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're thrilled to have you here, and we're looking forward to a fun episode today. To kick us off for episode 11, Brian is going to go over news and resources before diving into today's topic. Brian?
Thanks, Adam. We definitely have a packed agenda today with some interesting news and some special guests today. So looking forward to the discussion.
Yeah, it's an exciting slate we got going on today.
Yeah. The first item up for medical research news is on cure or remission for CAR T therapies and how we'll know. So this is, not quite a news article, but not quite a full paper, right? This was more or less an opinion piece in the journal Human Vaccines and Immunotherapeutics.
Kind of a response to something that was published previously.
Correct. And it's really sort of questioning and digging into the efficacy of CAR T cells in terms of oncology indications and what the success to date really means for some of the first CAR Ts that we tried out.
Yeah, it's a really interesting paper. It is something that is very close to what we've talked about many times, mentioned before about my thesis work being in chimeric antigen receptors. And so functional persistence really is a term that's very closely tied here to what this paper seeks to answer, whether we got a cure or a remission. And what that means is this cure persistent, present within the body, within the patient for a long period of time? And I remember back when doing that initial thesis research, there was some evidence of persistence for over a decade. But with some of the stuff that's come out, it's not always the norm here. And so what are we taking a step back? What are we looking at? And so the term MRD, minimal residual disease, are there cancer cells remaining but aren't necessarily quantifiable without high sensitivity assays. And so paper goes through and tries to ask the questions of what we need to determine to be able to get to, is it a cure? What were your thoughts?
Yeah, I found it very interesting from that perspective, and you summed it up perfectly. That's exactly what they're looking at. And in terms of the questions that they're asking, I don't know if it was even possible to get those types of answers when these therapies started, right? 10 years ago, did we have the tools needed? Did we have the high sensitivity assays to really dig down into MRD, either with flow or with next-gen sequencing?
Mm-hmm.
And I think that answer was either no, or it wasn't at least widely available at that time.
Mm-hmm.
So I'd be very interested if this paper, the questions that have been posed here, get revisited with modern research techniques.
Mm-hmm.
And I was also thinking of it in the context of how widespread this treatment is now, right? More than 7,000 patients have taken some form of CAR T therapy, and it's maintained a pretty impressive record of efficacy across a number of different trials and across the commercialized therapies that are out there. So
saw those numbers, and I was kind of curious because I didn't get a chance to look up this part, but is there a registry that collates all those numbers, or did they have to go through and add that all up?
There are a couple of very nice review papers that do collate all the patients and subjects on clinical trials.
Mm-hmm.
So those definitely there are databases and registries of. But I think it also references 34,000 patients worldwide.
Mm-hmm.
And to my knowledge, there's no registry of all 34,000 of those patients.
Yeah, really interesting. Impressive to see the numbers. As we know, it takes a lot to generate the therapy. Essentially getting the cells from the patient, modifying them, growing them, expanding them, putting them back in. It's not a simple therapy by any means. And so the amount of work to get it to that point, to those numbers, is very impressive.
It is. And then with the questions that they're posing here, it's all sort of very individualized therapy, right? Where this is all autologous CAR T at the moment.
Mm-hmm.
Are these questions, you think, answerable or generalizable? So even with all of our modern research techniques where we're able to get the high sensitivity to detect down to very low levels of MRD, or look at everything that's going on with cells to understand exactly how they're functioning with spectral flow and things like this. If we get an answer, will it apply to multiple patients or just the one patient that we get the answer from?
It's difficult, right? It almost makes you think of science fiction, and you just run a scanner over somebody's whole body to be able to identify if they've got some sort of disease or illness or something. We were talking about cancer cells. If there is persistence of just a few that could expand, you may not pick those up, even no matter how effective your really high intensity assays, the high sensitivity assays are. And so it's going to be a question that probably will remain unanswered for a while with that difficulty.
Sure. I mean, we're talking about MRDs with sensitivity of roughly 1 in a million cells as being sort of the gold standard at that point.
Mm-hmm.
And if you're only running 1 to 3 million cells, what are your chances of detecting a cancer cell? So it is
right. Within a body that's got trillions of cells.
Exactly. Right. And, that then bleeds directly into the second piece of medical research news I've got. So I brought up the paper, again, not exactly medical research news, but I found it a very interesting paper. And this one was published in Cytometry Part B. It actually came from the AAPS Flow group. And AAPS is the American Association of Pharmaceutical Scientists, a very influential group dedicated to bioanalysis. And this is the portion of that group dedicated to flow cytometry. So definitely a group that we're very active with, and always focused on sort of the biggest and best issues in flow cytometry today. And, towards our point on how difficult it is to detect some of these things and how applicable an assay may be in a new subject, this paper's on testing flow cytometry assays in rare matrices.
Yeah, really excited to see a paper like this come out. We've been working with H62 from CLSI, so it's the Clinical and Laboratory Standards Institute, for quite some time. It basically is the part of the core of how we develop validations and qualifications, making sure that we have robust standards around what we're doing in serving the clients there. And so this expands upon it, right? H62 is a great document, but it can't possibly touch every single avenue for a validation or qualification. And so here we're looking at the rare matrix samples. One of the things I found interesting was talking about validating these rare matrix samples and how you may even want to test on known samples, non-rare matrix, before going into the rare matrix. And we talk about this all the time, even if it's rare or not rare, when we're doing disease state versus healthy donor for a validation and just trying to get a handle on the performance and robustness of the assay.
Correct. And, if you test on a synthetic matrix like that, I'll call it synthetic, even though it may not be synthetic, maybe surrogate is a better word. Mm-hmm. You can at least get some parameters, right? Like if the marker is at roughly a similar antigen density, is my signal going to be on scale? Is the antibody binding or not binding? Is it specific within the context of at least this surrogate matrix? So it's not a full answer and nowhere near the complete answer that we would want. But in the absence of that matrix that you're trying to test on, it may be the best that you can get.
Right. Absolutely. Always talking about how we can best do it. Is it spiking? Is it buying samples, disease state samples, if that's an option? Many different ways to attack the problem, but realistically trying to get to that answer as close to what you're going to see in the clinical trials. I did see too, they were talking about cluster sizes here and number of events within the abnormal phenotypes. We were talking about low event samples, and it's always something that's kind of a sticky wicket, right? You don't want to say, well, we need 50 events to be able to tell you this. And so that's where you do your LLOQ, also your LOD, to really get a handle on where it is that signal is reliable.
Right. And, you made a good point that there's no guidance that can sort of carry you through each and every situation, especially in flow cytometry. I think about it as being on an island surrounded by water, and you've got to just keep putting rocks from the island out into the water until you can build a bridge to wherever you want to go. So, I see this as sort of the first rock that we're putting out there. And then the next things that would make sense are how to do in-study validations in a regulatory compliant way. And then also, how much you can get from each sample and how to do sort of a validation in series, if you will, as opposed to having multiple samples to test all at one time.
Right, because in trial validations is so tricky because when would you be doing something like that? It'd be a couple of issues. Maybe you need to transfer the assay. You had it in-house, but you can't handle the number of samples, and so you have to get it to a vendor, and you want to be able to make sure it works on their machines, and then you have to do the validation, but you can't stop the clinical trial necessarily. But they're fresh samples continue to come. It could just be change in instrumentation, something as simple as that, but that's just a simple test script. And so, yeah, it would be certainly something that would be beneficial to have those standards around.
Sure. Yeah, and I think about like AML samples or AML bone marrow or CSF samples and things like that, things that we wouldn't be able to get from other patients, and then you basically had to take the first 5 or 10 or 20 samples from a clinical trial and use them as basically the test set, and then potentially exclude them from later analysis.
Right. All comes down to getting the proper permissions too at that point. And it's such an involvement of different stakeholders. You get the clinical team, the collection sites, you've got the sponsor, usually a big pharma or pharmaceutical company. Then obviously you've got the CRO, the companies that are doing the analysis. And so it really is a dance, right, to be able to get this to work effectively. And the companies that do it the best have really built up their communication and work with one another effectively there.
Yeah. But, the mark of any good paper is that it brings up even more questions, right? So I think definitely this one starts an interesting conversation and hopefully more to come there.
Absolutely.
Yeah. So I guess we'll transition right over to more on the investment news side. Of course, the biggest news in the past week here has been the negotiated prices for the Inflation Reduction Act.
Yeah, this was interesting because as I'm starting to read the article, there's a lot of sunshine and happiness and, saving money. And then on the other side, it seems like there wasn't such an agreement as to how beneficial it's going to be. But, you're talking about the Medicare government negotiating with pharmaceutical companies. And I'm sure you can't expect that everyone's going to come out super happy. But I think in the end of the day, the point is that therapies can be extremely expensive, and you don't want those therapies to be prohibitive such that people can't get what they need. And so the goal here was really to try to bring those costs down. And so it looks like it covers about 50 million people. The expectation is taxpayers are going to save about $6 billion in 2026. Patients will save about 1.5 billion the same year. And this is mostly around seniors. And so, people who have generally passed their prime earning years really can use that support.
That's it. So, the intent of it is always good, right? To bring down drug prices. I think to your point, it's always good when both sides can come out saying that they're either happy with or can live with the outcome. So we didn't see an immediate drop in pharma stock prices, nor did we see an uproar from patient advocacy groups saying that this is a terrible thing. Hopefully it will reduce the tax burden in the U.S. And hopefully it will make it so that it doesn't completely upend the pharma business model there. But definitely time will tell, and glad that this first round of things didn't rock the boat farther than anyone thought it would.
Right.
Yeah. And then in other sort of reimbursement news, I also have on here the autologous cell therapies in practice. So Bluebird Bio reporting out some financial results.
So this is really interesting. We're talking about sickle cell, which is an inherited blood disorder. Probably most, if not all of the listeners, are familiar with the fact that it reduces the ability to transport oxygen. And so it can also cause extreme pain at times. And so it's an unmet need. It's something that there's not a ton of treatment out there. And so getting gene therapy, we've got two different ways of going about it. Casgevy is CRISPR-Cas9, taking cells out, modifying them, putting them back in, increases the expression of fetal hemoglobin, whereas Lyfgenia is transduced by lentivirus and then produces protein similar to hemoglobin A. And again, it's the same thing. We're taking the cells from the patient, modifying them, and reinserting them.
Exactly. And, both different ways to modify cells and then sort of address an issue there. And very similar to CAR T, where, again, the autologous nature of it is the challenging piece from a commercial standpoint. Mm-hmm. And I feel just a great kinship between the commercial challenges there of doing autologous cell therapy and the commercial challenges of doing flow cytometry, where we both have to, wait for whole blood samples to come in, and then each and every patient is a pretty significant impact on, what we're doing on a regular basis.
Right. And so with this, looks like we've got 4 patient starts so far, U.S. only for Bluebird with Lyfgenia, so far 20 for Casgevy. And interestingly, these kind of numbers, I guess I don't want to say wreak havoc, but definitely have an impact on the stock prices for the companies. They were talking about Bluebird dropping about 18% with the comparison of the numbers, but they do expect to be ramping up very significantly as the year moves on.
That's it. I mean, with these neither one of these are small, but with these smaller biotech / pharma companies, definitely the performance of a single molecule can have a pretty significant impact on their overall value there. So interesting to see how that autologous strategy plays out in practice. And best wishes for both companies moving forward here. Hopefully they can continue to offer these absolutely breakthrough therapies that are changing the game of what's possible with medicine to more and more patients.
Yeah, absolutely. Like I said, unmet need. And so anything we can do to start helping more is definitely beneficial. So what do we have on tap for today as far as the main portion of our discussion?
Well, transitioning into that main portion, we do have Natalie Carroll in KCAS News. We've got Natalie Carroll, our scientist 3, who's fresh out of her trip to Australia to do the Cytek training there, is now on her way over to Lyon, France, literally as we speak, I believe.
I think so.
Yeah. And once she's there, she'll do the exact same procedure she carried out in Australia. So that way we've got the same analyst on all three instruments on the same validation. So definitely a non-traditional way of doing that, but the best way to ensure harmonization between all three sites.
I got to say, just as a complete aside, I have this image in my head of her having one of those classic brown suitcases with, the sticker for Lyon, the sticker for Melbourne. And, she's earning all of them so far.
All right. I see the next employee engagement gift there. Definitely.
But yeah, I mean, I just wanted to highlight too, Natalie was picked for this because, she's a subject matter expert with Cytek and, has really demonstrated strong proficiency there. And so putting our best foot forward to make sure that everything is done as it needs to be, and excited to see her knocking this out.
Yeah, and just to recap a little bit from our last episode, the reason that we're doing this is because so many trials now are going global. We mentioned the difficulties of finding patients, enrolling patients in these trials, and sponsors are really having to go wherever they can find the patients. So that means having sites in multiple regions. So we, of course, are in the U.S., but we need to go into Lyon, France. We need to go into Melbourne, Australia, and make sure we offer the latest and greatest techniques to be able to analyze these things and get the answers to those questions. Like we mentioned in the initial investment, like those answers weren't possible earlier, but now that they are, how can we go out there and help our pharma partners to do that?
And for a long time, we did have not only the contingent in the U.S., but it was always a goal to expand out to the APAC region, just for what you had said, the additional coverage, right? You want to be able to see the variability throughout the globe. And if you're only testing in one site, it minimizes what you're learning in that study.
Yeah. And, to talk more about the point, then we can introduce our sort of main guest here for the main portion of the podcast. So towards that, I'd like to introduce John Bucksath, the CEO of KCAS Bio, and Stefan Cross, the CEO of Crux Biolabs. John, do you want to give a quick introduction?
Sure, absolutely. Well, first of all, Brian, Adam, thanks for inviting me onto your Conversational Flow podcast. Big fan of the output of you two gentlemen, and just thrilled to be here today. My background, CEO of KCAS Bio. Been with the organization a little over 8 years now, and just been real thrilled to be able to support the organization and really expand the organization and really be able to support global development at this point. When I initially started, we were very focused, very narrow offering in the bioanalytical services and traditional aspects like mass spectrometry and LBA services. But today we are a complete operation, again, both from a geography standpoint, but as well as a platform standpoint. Obviously today we're here really focused on the flow cytometry and the incredible development impacts that this platform has had on drug development and will continue. And it's also one of the most interesting ones as it continues to develop in its complexities and the challenges in handling these type of samples. So again, thrilled to be here, and I look forward to the conversation.
Thanks, John. Stefan?
Yeah, hi, Brian and John, Adam. Thanks very much for inviting me onto today's podcast. It's great to join you. And looking forward to explaining a little more about the alliance that we've set up with KCAS and Crux Biolabs down in Australia. There's a lot going on in the Australian clinical research space, and together I think we can really support many of our clients around the world now with a much stronger opportunity to support their studies from phase 1 all the way through to phase 3. So to give you a little bit of background about myself, I'm relatively new to the clinical research space. I joined Crux just over a year ago, but before that spent my entire career in the pharmaceutical sector. And a little bit like this conversation, it's been a bit of a global journey for me. Spent some time living in the UK and working across the European continent for several years. Moved back to the Asia Pacific region for several years, and then moved over to the US and headed up a US-based pharma business for a few years before returning back to Australia. So I'm kind of feel like a bit of a flow expert flying around the world. So look forward to today's discussion as well.
All right, excellent. And thank you both for being here. So to start out discussion, really just to continue on that vein of the importance of sites in all regions for these global clinical trials. John, could you give us a sense of, what sort of feedback we're getting from clients and, what we're trying to achieve with this expansion in Spectral Flow?
Absolutely, Brian. Well, first of all, we have been offering these services for a while. We had not, obviously, at that point in time, reached out to be able to offer the APAC portion of the business, but we did an in-depth study. What is the market looking for with rapid changes and advancements in technology, continuing development, and the evolution of how to actually do these studies and the complexity that they bring. We really wanted to hear from the market, our clients, and really try to understand, what is the need with, again, the kind of advancements that we're seeing. And one of the things that came through loud and clear from the industry was that there's this quantum shift moving into the spectral flow needs and expectations. But with that comes the complexity of making sure that you, one, have the resources to be able to do that, most importantly, the talent and expertise. And then from there, the commitment really from the organization to be able to offer that, to really support the drug development. It's one thing to be able to have expertise at a site and be able to do that work, but it's a whole other thing to have that expertise at multiple sites, harmonization, commitment from the organization as the business to also be able to offer that in a way to really help our partners develop drugs. And that's where the complexity really comes in. And I know we'll get more into that today, but the one thing, again, it really came through loud and clear is that there's an unmet need in the marketplace. And we felt that we had so much of the expertise, capabilities, and commitment from an investment standpoint. The one thing that was missing, of course, was our offering in Australia. So we were absolutely thrilled to be able to make contact with Stefan and learn more about his team, and we're just really blown away by the expertise and the professionalism that they bring. And we were just so thankful and hopeful at the time that they would continue to or consider partnering with us in this journey. And here we are today. So very excited, could be more excited actually about the opportunity working with Stefan and his team, and having Adam and Brian lead this along with the Crux team. I'm sure we're really going to make a difference for some of our partners, our clients out there, in really helping them develop drugs. And at the end of the day, that is the ultimate goal. We all have to run our businesses as necessary, but really helping to expand the expertise, the science, and help develop drugs. At the end of the day, it should, at the end of the day, work out best for everybody, the CROs, the developers, the innovators. So just very excited about everything that's going on here.
And John, going to your point about spectral flow, I mean, it really has come a long way. You heard all of it internally, right, from Brian and I clamoring for moving in this direction, even just at the Philadelphia site. But then, as you mentioned, doing the research to get it expanded to the other sites here. And so having it at Lyon, having at Australia. And this is something where back in 2017, we had a demo of the Cytek Aurora, and have been pining to get it on site, and using it ever since then. And so, for me personally, it's really been exciting to see us go in this direction, specifically just because of what it can do. Conventional flow is a very strong tool. However, just can't query as many parameters as you can with spectral flow. It opens things up for you more. Other things that it can do, such as autofluorescence subtraction, small particle detection, and so really has something that we needed to add to our toolbox, and excited to have the team that is as capable as they are in executing it as well.
And I will second that. So the Crux team has been just an absolute pleasure to work with. The Flow community is small, and generally sort of sticks together as much as possible. And as soon as we started working with them, we knew they were an integral part of the team, and an excellent thing to then add on to the operation overall, in terms of the partnership. And then in terms of our selection of Australia specifically, with those sites, Stefan, can you give us a little bit of insight on why Australia has sort of come to the forefront for a lot of clinical trials?
Yeah, of course. I think there's a fairly well understood market here in Australia that it's very well geared towards supporting phase 1 clinical studies. And if you unpick that a little bit and understand a little bit more about why, one of the fundamentals is there's no requirement for an IND in Australia to commence a phase 1 study. So for sponsors, that could be saving 6 to 12 months of their time. The ability to rapidly start up a study here in Australia as well could have someone in a clinic within 5 to 6 weeks from study initiation, with all of the infrastructure and the processes that are built around supporting phase 1. And importantly, the data that's generated in Australia is well recognised and accepted globally. So, of course, if you're operating in a tier 1 market, particularly if you're a US or a European sponsor wanting to accelerate your phase 1 program, you can be assured that the study data that's generated down in Australia is accepted and can be part of your overall clinical program. And there's another advantage which some companies take an opportunity around, and that is that there is a 43.5% tax incentive from the Australian federal government for eligible R&D spend. So it can actually make your R&D spend spread a little further through that program as well. And all of that has created a really strong ecosystem, as we call it here, across more than 50 sites to be able to perform clinical research. All of the scientific expertise that's generated and initiated through our university infrastructure, all the way through to the commercial support and the CROs that exist here as well. And I think overall a fairly well accepted and strong reputation for undertaking clinical research in Australia, particularly from our US clients that have seen this for several years now.
Appreciate that, and definitely second the point on the importance of trustworthy results, right? That basically when you get results from a site in Australia, then they usually go over very well with the FDA or the EMEA. And in our case specifically, since we're harmonizing these assays across sites, we're gearing results for that exact purpose. So yet another strong point in the partnership that we can then make sure any results that we generate will be accepted anywhere in the world. Towards that point, and in terms of speeding clients through the drug development process, we always like to keep both the patient and the sponsor as the key focal point of everything we're developing. We're doing this for them to get drugs through as quickly as possible. So John, if you can comment maybe on what the most important things are to keep the same in between sites, or what we're doing to basically take the headache out of carrying out these multi-site trials.
Well, Brian, I'll be the first to say, I'm not a flow cytometry expert, but being in the analytical sciences my entire career, over 35 years, it really really comes down to, I call it the three big buckets, right? There's going to be a lot of, obviously, to try to control any biological variation between samples in any form that we can. I know that you've talked about that before and how we do that. Obviously, the instruments, any fluctuation or issues or variances in the instrumentation have to be weighed in. And of course, as we expand that in multiple geographies, multiple analysts, you can see that the compounding interest of some of that variability has to really be considered, and how detrimental it could be to really the assessment of the data that comes out of these studies. And then really the other aspect, really sample processing by different analysts and how they do that, and the techniques. So many of the parameters for all the types of platforms are very similar in the variability. I just think for flow cytometry, since it is so specialized and the data is so complex, it just really takes on a whole new level of complexity to focus on keeping the variability under control, demonstrating that you have the processes, procedures in place, and commitment in investments to make that work across multiple geographies. Like anything, when you start to add those up and multiply the additional aspects of that, it becomes quite complex. So I've been incredibly impressed by the team's commitment to this. It is taking several weeks and months to get this design set up, confirmed, as you mentioned. Natalie has been quite the trooper, traveling the globe here to really, justify one of those variabilities, really taking the time to do that, and expenses and commitment to make that work and demonstrate that we take this very, very seriously. And that's just one of the parameters. So again, same type of equipment, same type of processes, same type of SOPs, protocols. There's just so much that goes into it that, at the end of the day, it might look simple per se, and it sounds in many cases like, well, absolutely, that's what you should do. But actually executing that, designing it, setting it up, and doing it well is really where it will make the difference. So I'm convinced that we're, setting the stage for really that next big run in offering global flow cytometry, thanks to the leadership of this team, and just really look forward to continue to support that between all the sites.
Right. And John, a great point. It's going local to global. And a lot of the processes that you may have in a local site, you have to think about how you can translate that so that it's consistent at other sites as well. And so a lot of communication has to happen up front, to your point about weeks and months of preparation, so that it can be done properly. You don't want to transfer an assay, but then the things that have supported your assays all that time that have made it reproducible aren't transferred. And so it all has to go as a big package there.
Absolutely. Just, SOPs. It's been had the opportunity to sit in with the quality groups when they're going through and harmonizing the SOPs and making sure that when they read a sentence that they interpret it the same way. And if you've ever gone through that exercise, which I'm sure many of us have, and most of us here obviously on this call have, and I'm sure a lot of the viewers have probably experienced that as well. What is that SOP? What does that sentence, that SOP mean to you, and how would you execute it at that point? So the teams are really, really digging in and making sure that it's going to be the measure twice, cut once type approach. That's going to make it obviously a much easier as we embark on developing and working with our clients to produce the data that they're looking for. I think one of the things that's worth mentioning, and I'll throw it out here, is just even how we're consolidating the data together and really having that managed, cohesively. So I think maybe that's something that Adam, Brian, maybe you can speak to as well.
Sure. And especially with flow cytometry, where the analysis is definitely complicated. Some people can call different parts of it subjective, which I would of course argue against. But definitely, no one can deny that it's certainly complicated. So having a central reviewer is absolutely critical to implementing high complexity panels in multiple sites. And that's definitely one of the pieces that we were most adamant about setting up quickly and first, to make sure that the data that comes out will get treated the same no matter which region is generated in. And ideally the SOPs and processes will be the same. That way we can trust that the data that is generated will be the same, and then the interpretation is the same. So then we can make adequate decisions on the back end of that trial.
I think jumping into what you just said about subjective there, you may have just spawned another podcast topic.
Oh, no.
Because we can really talk a whole lot about, analysis of data. Where do we place gating? The iteration of the gates in the hierarchy, and what does that all look like? It is subjective, right? But what we're trying to do is get down to a core so that it is consistent and done the same from place to place, and certainly moving in that direction. One of the things I'm curious about, Brian, I know you had put this on the agenda, and you briefly mentioned to me last night, is this duck mindset for harmonizing. And so do you want to expand on that a little bit?
Yeah. So what is a duck mindset? I think of it as, when you look at a duck just swimming across a pond, it looks pretty calm, right? You just see the duck and it's seemingly floating with nothing really happening. But if you look under the water, the duck is paddling as hard as it possibly can, right? So they are working like hell underneath the water where you can't see. The effect is very simple. So if you think about it from our sponsor standpoint, basically they see just the duck floating across the water. So they talk to us and say, Adam, Brian, John, Stefan, we want this flow cytometry panel, and we want to run 10 samples in Australia, 10 samples in France, and 10 samples in the US. Give us a call when it's done. And then they just leave us with that project and we go do it. And they don't see all of the different work that goes into making sure that those 10 samples are exactly identical in all three places. But if you think about the alternative, they would have to find 3 labs, ideally with the same instruments, that maintain them in the same way, that have SOPs that are roughly the same at least, but without a QA review and real formal gap analysis, you have no idea how close they are. Towards John points on different interpretations of sentences by QA teams, and then who knows how that goes once it gets to the lab as well. And then they're analyzed differently, and then so you get all these different results that may have almost nothing to do with each other. And then you compound that with the complicated analysis, and it gets very, very difficult. So thinking about how that looks from a sponsor site, if a sponsor was to try to set this up themselves, how many people would they need to manage the different CROs or groups or investigators that are doing this individually, and then assemble that into an equivalent data set? And that answer grows quite rapidly there.
Yeah, and it is one of those things that, we try to do, make it look like it's easy, despite the fact that there's a lot of work going behind the scenes. Sometimes I would say maybe to our detriment that it seems so easy. Why can't this just be done tomorrow, when it generally takes 2 weeks or whatever? So I certainly appreciate that navigating. Very curious too, then, Stefan, from your viewpoint, having been involved with the transfers that have been going on so far, your thoughts on the experience, any challenges, and what you've learned there?
Yeah, Adam, it's been an amazing experience for our team. Obviously, it's been great to have the background and support of the KCAS team. But one of the other things that came out in the earlier conversation when you were talking about it, John, was Cytek has been an excellent partner in all of this as well, and they've been highly committed to working, particularly through KCAS and obviously onto us as well, to make sure that the equipment at its core is harmonized from the factory. And that's one of the things that we've noticed. It's been a real white glove service with them, bringing, installing, qualifying, supporting our team on site, and doing exactly the same thing, as I understand, with your team in Lyon as well. So seeing it all the way from the equipment supplier and understanding and really working with those guys has been the first foundational part. The next part is obviously working really closely with the KCAS team. And all of this is only going to be possible if we've got a team that are willing to want to work together. And as John mentioned at the beginning, this has been a partnership in several months of making, and across that journey, we've really got to know each other really well. Very professional teams on both sides of the pond, and in doing so, what we've learned is that we've got a really strong alignment around wanting to deliver a highly aligned, globally relevant service in Spectral Flow that is, it seems, a very rare offering these days to be able to provide effectively 3 regions through 2 companies under one highly harmonized approach. So what we've put together in the last few months, I think, is a pretty phenomenal offering. I'm pretty excited about seeing what we can bring, not only from perhaps the initial phase 1 work that we're going to be able to support, but ultimately Crux's involvement in supporting phase 2 and phase 3 programs as projects expand globally, and as the KCAS team look to support projects that are expanding globally and are looking for an APAC and an Australian arm of their study. So, Adam, it's a pretty exciting time.
I would certainly agree, and on all points, with Cytek, been very helpful in getting the instrumentation aligned there, harmonized. Really appreciate how close you can get them compared to standard flow there, and I think that just aids in our ability to expand in this way and really provide top shelf data for the clients and really drive their studies forward. Brian, from your perspective, thoughts and learnings?
I'd say one of the best things that we have done and are continuing to do, and are going to actually put out a number of white papers around, is our quality alignment between sites. So our ability to support regulatory audits at each site, and then be able to describe processes occurring at other sites there. John mentioned the global SOPs as a standard process, but then also, our processes around the study driving documents. So single analytical plans, single, basically central data review for all data, really gives regulatory agencies and sponsors to date a lot of comfort that the entire data set is being treated equally and in a very thoughtful and considered way, in compliance with all regulatory guidelines.
The one thing I'm not hearing here that I at first would have expected was a big challenge is, how do you get a site in Philadelphia, a site in Lyon, and a site in Australia all on a call at the same time? But I think that's probably been the biggest hurdle. Everything else has been met with resolve, and everybody's rolled up their sleeves to get things done. So not much that we can do about time zones there.
I think, Adam, Australians have found it pretty normal that we need to accommodate the global time zones. So whether it's 6 o'clock in the morning or 10 o'clock at the night.
Mm-hmm.
Well, if you're in this business very long and you have any certain amount of responsibility within a CRO at a leadership level, oftentimes that's when you get to catch up, is either before the day really starts or after it's done. So I think it flows right into a lot of the common practices, if you will. We work very, very hard to try to take care of our clients, and we're doing that during the normal business hours. But I couldn't be more impressed by the professionalism of this team. And Stefan, your team has just really made a huge impact on the impression, not knowing exactly where we're getting to, but obviously working with you and being very impressed. But just Natalie, having a chance to speak with her in Philadelphia just last week. And, I just took the opportunity. How was it working with the Crux team? And she just kind of stopped, looked me right in the eye and said, phenomenal. They're absolutely phenomenal. And when somebody can say that's traveled and is going through the processes, and quite frankly, we really challenged her to make sure that she's, responsible for actually the execution. When somebody can say that, having the opportunity to meet face to face for the first time, it just gives a lot of confidence that we're really prepared and well suited to be doing this work.
Yeah, great feedback. Thanks, John. And Natalie did an awesome job, and I think it's a great program design that you've put together as well, that you're now having Natalie attend to the Lyon side and support them, and some of the learnings that I'm sure she's taken from Crux will then pass back onto the Lyon team, and then from that trip back to the Philly team as well. And that's part of the great thing about this alliance, is that it will be continual learning across our teams. Every time we pick up a project, I'm sure there'll be something more that we can improve as we go.
Absolutely. I just want to take a moment here to maybe embarrass Adam and Brian a little bit, but their leadership has been phenomenal in setting this up, and their passion for it has just been contagious. So anytime you have that type of commitment, just really, really excited to see how this evolves and grows over the commitment over the next several years.
Yeah, I really appreciate that, John. And, I was thinking the same, going back to you, since we've been with the company. I mean, you've brought on the Philadelphia location, you brought on Lyon, worked on getting the partnership with Crux. So I know there's been very little downtime on your end, and the hard work is very impressive, and the results as well.
Absolutely. And, as we've all said, definitely we grow through new projects and challenges, and hoping that we find some more of those. We'll actually be going down to the Australian Biotech Conference at the end of October. So looking forward to meeting up with the Crux team in person there.
Yeah, can't wait to have you guys down.
Yeah. Indeed. All right. We are getting close to time, so I'll go ahead and read out a little bit of fan feedback, and then we'll go into a little bit of a closing session. So in terms of feedback, our question from a few episodes ago is, what is your favorite type of automation and why? And we had sort of two categories of response. And the two categories that we got, one sort of group of people seem to be very interested in the Echo and Curiox platforms. And I found their feedback interesting because they all were flagging the ability to miniaturize assays and basically do the same type of testing, but now instead of using a mil of blood or 2 mils of blood, potentially get down to 50 microliters or maybe 100,000 PBMCs, and do the same type of staining steps, but just with a very small volume of blood.
It's important, right? Because samples are at a premium, and you're taking these samples from patients that are ill. And so any way that you can miniaturize the assays really is a huge benefit there. Also, too, you then lengthen what that sample can provide to you, whether or not there's more assays that can be run, other things that can be done there to gain more data.
That's it. Why stop at 40 colors if you can get 2 assays and do 80 colors, right? So, always that.
It only have to be flow. There can be other assays. There are more out there, right?
Say, what? What? Oh, yeah, sure. I guess. Fine. The other category of automation that we had respondents talk about was the TCAN system. And I put this into basically the group that had sort of established and broad ecosystems of automations that they could fit into. So then the TCAN would fit very well into like a flow group, flow automation workflow, where maybe it wouldn't be just the master mix prep. It might be managing the samples through a process. As you say, I understand that there are some other types of tests that you can do outside of flow cytometry. So I assume that's what they might be referring to. But yeah.
I mean, the TCAN has broad applicability and has been used for a good deal of time. And so certainly I'm more familiar there with that than the Echo and Curio, just in the roles that I've had. But automation is certainly a direction that we need to adopt more and more as you want to drive consistency. But you have to make sure that those units are suiting the needs. And so, Brian, generally, when you're looking at automation, automation, what are some of the things that you look for?
Exactly what you say. Like, we've got a workflow now. What are the important points of that workflow? So do we need to have a certain volume of blood? Do we need to have a certain CV? And how will those instruments fit in and either improve on or maintain the critical parameters of the workflow?
Mm-hmm.
Whenever you're thinking about automation, you don't need to think about can it replicate what we're doing exactly, because you'll have to think about it differently. But as long as you keep the critical parameters in mind, then you can design a new workflow to fit that automated process better.
Mm-hmm. Agreed.
Yeah. In terms of questions to hear back from our guests on for our next podcast on sort of sample prep. And this one is, which anticoagulants or fixatives do you prefer for flow assays, if you have a preference, or which do you find sort of most broadly applicable?
So you can't just say whichever one works best?
No. We want opinions. We want controversy. There's a podcast here, Adam. So we'll see what we can get.
I just want to be agnostic. Whatever works best. Let's test it out. Let's make sure that it applies to the sample type, where it's coming from, what we're trying to do with it. There's so many parameters that go in. Yeah. So we do want to hear some. We want to have some flavor and some color here. So please don't be boring like me.
All right, and that brings us to the end. So as we wrap up here, we did just finish the Olympic Games, so very, very appropriate for a podcast on global flow cytometry. Lot of different countries. Kudos to all the athletes, just incredible performances all around. But did want to ask our guests what stood out to them from the Olympic performances.
Well, I can start. You don't want me to start with the amazing breakdancing, do you? That's probably the one place I could start. But perhaps I'll actually start with the overall medal tally for Australia. I think we finished in 4th position, which is a pretty phenomenal effort for a country of just under 30 million people, or 26 million people. So we are a pretty proud nation of sporting activity, and we love the Olympics because we get to showcase the great sporting prowess of Australia, and particularly in the swimming pool, where I think we compete pretty well head-to-head with the US team.
Nice. And did you want to comment in any more detail on the Australian breaking performance?
I think that's enough said, isn't it?
Okay, fair enough.
And a lot of ink spilled on that.
I'm actually intrigued how global this event has gone. When down here in Australia, it certainly got a bit of airtime, but I didn't expect the level of global infiltration of this particular event. But I guess anyone can be famous for something, and that's what.
And that's the amazing thing is you can be amazing at what you do, and you don't reach the level of fame as somebody else who has a moment like this. Seen some of this, with what was it, national anthem singers or whatnot, where all of a sudden it just pops at just the right time. And maybe the wrong performance. I don't know. It seems like the right performance. I don't know. But you're right. Yeah.
I think moment is a good descriptor. Well done.
Mm-hmm.
And John, what was your take on the Olympics?
Well, from my perspective, being an old basketball player, I always had a lot of pride in that the U.S. Formerly would send its best college players and still walk away with the gold. And of course, as the world became more competitive in basketball, we started bringing in our professionals as well. And of course, there was a time when the U.S. did not walk away with the gold, and I thought maybe we were getting ready to see that again when I believe they were playing France.
Mm-hmm.
And I really thought our Lyon colleagues were going to have some bragging rights there, but they pulled it out, kind of at the last minute on some phenomenal play by Stephen Curry. And, it seems like some unorthodox shots, but somehow he makes them routinely. But yeah, I think that, and I think the one thing that really stood out to me though was the pistol shooting. The gentleman from Turkey and just kind of walked up casually and took silver. To me, that just intrigued me at that point in time. So it just goes to show you that there's a lot of talent out here. One more statistic. I'm an analytical guy. That also stood out to me in I believe it was 1984 when Carl Lewis was the fastest man in the world. Comparative to today, the 100-meter dash, that same time, he would have been in last place.
Wow.
So it just kind of goes to show you how the competition continues to increase and performance continues to increase. Not unlike our business, right? Expectations continue to grow, technologies continue to grow, and we have to keep up with that. So.
And to your point, John, bragging rights. I mean, we've already dealt with that as a company between when we had the Super Bowl between Kansas City and Philadelphia. Didn't really go the way us Philadelphians would have hoped, but Kansas City is a great team, so it can't really say anything. And then, from a broader context, you're mentioning about competition within the CRO space. I mean, bragging rights, bringing Spectral across the globe, and there aren't many companies that can say that.
That is true. That was one of the things that really stood out during the evaluation. Obviously, there are those that do offer it, but we were surprised, I guess, that the minimal minimal competitors out there that are actually doing this. Couple that with the timing and the feedback from industry, it seemed like definitely the right thing to do from an opportunity, and again, to continue to support our clients in developing their drugs.
And the other just brief topic as we close out is fun with speech to text. So we talk about technology increasing the flow cytometry field all the time, but it goes in all directions, right? So speech to text has been an upcoming thing with Siri doing her best to listen to us and do what we want, and now a number of other different chat assistants. Wanted to get your take, John, maybe we'll start with you on, have you ever had issues with speech to text, and any good stories there?
If I didn't know any better, Brian, I think you were setting me up from some of the team. Siri and I have a love-hate relationship. I've promised my daughters I will never text while in a vehicle, so obviously have the audio, and I am absolutely convinced that she takes great pleasure in manipulating words as I send those texts. So I'm sure you have been a victim of one of my texts that have come through that I promised that I was going to do verbally. But rest assured, I've tried many, many times to revamp the versions of that text, and she just simply will not cooperate in putting the right word. And there's quite some humorous stories from the team about some of my texts, but I'm glad I can, for lack of a better word, provide some pleasure at my expense on my texting capabilities through Siri.
Happens to us all. I was complaining of, whenever I try it, then, almost without fail, I'll text something to my mother, or my wife or something, and it'll be something like, oh, did you pick up the butt? And she's like, who are you trying to text, and what about? Like, what led to this misunderstanding? So yeah, I've essentially tried to swear it off as much as possible.
So it does make me wonder, though, what sort of an accent does it use as its baseline? Because depending on where you're from, everybody has an accent, right? It's just a different accent. Now, for me, I grew up in New York, and so going to Penn State, my New York accent is kind of watered down in normal speech. But as people who know me can attest, if I get worked up, all of a sudden you're starting to hear my New York accent. And I've tried to text using Siri with my New York accent, and all it does is make me more worked up. So I've gone away from that direction. I agree with John. I might have more of a hate relationship. I tend to just avoid texting when I'm in the car and just wait until I get out of the car.
I think the safest way to use this is to make sure if there was some kind of automatic auto disclaimer at the back end of voice to text. So it actually appended to the end of the text message. This was generated by voice to text, so that it helps us cover our tracks a little bit with these mistakes that obviously are quite inherent in speech to text.
Absolutely.
And Stefan, does speech to text work okay with Australian accents as well, or how does that function there?
Well, I've tried them all. Yes, it does. Siri's not bad. I do have a Tesla, and it is terrible. It only has American English as its default, and it is the worst speech to text. So I've completely given up on using it in my vehicle.
Fair. At some point, you just have to, declare the technology is just not quite there yet.
It's not.
Yeah. Luckily, not the case for us. And, that will, I guess, bring a close to the episode for today. We do have a very exciting recording next month, and we're going to have some more guests on from the software side of the house on how to get the most out of high-dimensional analysis. So we'll have some guests on from Dotmatics to talk about some of their new systems there.
And I do want to point out, Brian, because you made this a thing recently, one of our podcasts, maybe the last one before. We actually haven't talked about food this podcast, so new ground for us.
I know. Yeah, I mean, we lead the podcast. We're not hungry. Mm-hmm. So, yeah, I kind of like this. Well, let's see.
Different time of day, right? So.
Yeah, there's that. All right. Well, thanks, everyone, for joining us, and we'll look forward to talking to you on the next episode.
Yeah, and thank you both to our guests, John and Stefan, for joining us. Really appreciated all the insight that you provided and enjoyed the discussion. Thank you.
Great chatting with you guys.
Thank you, gentlemen.